After uh, protocols, after going through the procedures, so the next thing is for you to see the results of the experiment that I did on your behalf. So just an overview. Remember the substrate for each of the experiments. For example, the carbohydrate is the starch. The enzymes that we're looking at is the alpha amylase, which is present in the pancreatic juice or extract. And the end product of the digestion should be maltose for starch. Then for the protein, we use the BAPNA, which I said is the synthetic polypeptide, and it's supposed to be digested by the trypsin, which is present in the pancreatic juice, and the end product is supposed to be the amino acids. Then for the lipid digestion, we used milk, and we said that we combined bile salts with the pancreatic juice, which contains the lipase. So the bile salt is meant to emulsify the fat in the milk by breaking down the large fat globules into smaller globules, and in such a way it's kind of increase the surface area of the fat molecules so that they can easily be digested by the pancreatic lipase. And the end product should be monoglycerides, or you can also say glycerols, and the fatty acids. And so I already put this in the overview and to tell you where you have the stats of each of the nutrients, the digestion of each of the nutrients. And you should take note of this, especially for your quiz and for your exam. So we can ask you, where do you think the digestion of fat starts from, or the digestion of carbohydrates, or digestion of protein, and the different enzymes, and like the pepsins in the inactive form, which was the pepsinogens, and the role that HCl plays and things like that. So take note of that when you're studying. All right, so we already mentioned this in the video, and so you're supposed to have 5 tubes, and they're supposed to add each of these reagents to each of the tubes. And at the end of adding all the mammalian ringers, the dairy water, the starch, and the pancreatic juice, you're supposed to rotate and you cover them with the paraffin. Remember, the 5A or 5C is usually the positive control, and you usually add the enzyme directly to the starch. And after that, you incubate 2A or 2C in ice baths, and while you put the rest in a water bath at 38 degrees centigrade for 60 minutes. And after that, we're supposed to run a test. We're going to use 3 acids. We use the potassium iodide to test for starch, the Barfoed to test for the saccharides and monosaccharides after the incubation, and the BENEDICT'S assay is used to test for the amount of reducing sugar that is present in your samples, which are the test tubes after digestion. All right, so after incubation, what we're supposed to do next is to test for the digestion of carbohydrates. So we're supposed to do 3 tests for this purpose to determine if we still have carbohydrates in our samples and the type of carbohydrate that we have. So the first one is called the potassium iodide test, IKI, and what you're trying to do is to detect for starch. So if you have starch, it means that there's no hydrolysis, and if you don't have starch in there, it means that there is hydrolysis. So now what you do is to get a depression plate, and you're going to label the spot. So it usually comes with a well, like a concave depression, and you're supposed to add a drop of each of your samples from the test tubes that you've incubated into those spots that you've labeled, and then you add 2 drops of the IKI on your sample, and you check for if there's any color change. So if it's orange color, it means that your sample is negative for starch, and there's hydrolysis that's must have taken place. But if it turns blue black in color, then it means that there's still starch there. That means it's positive for starch, and there's no hydrolysis taking place in your sample, even though you've incubated them at the optimum temperature. Then the next thing you need to do is to test for the monosaccharide and disaccharide in your sample. So the Barfoed is used to distinguish between them. And then what you do is to get a separate test tubes, so 5 test tubes, you label them. Then you add 5 ml of the Barfoed solution to them. And in addition to this, you add 1 ml of each of your samples into them and you mix them. And then you put in the water bath, 38 degrees Celsius for 5 minutes. And if you didn't see any precipitates, even though the sample is positive for IKI, it means that you still have starch in your sample. There's no monosaccharide disaccharide. But if you see that your samples at 5 to 7 minutes after incubation or during incubation, it turns orange, red orange, then it means that you have monosaccharides in your sample. So if it turns, it gives you like a marked reddish brick red precipitate, then it means that you have the saccharides in your samples, and usually see this around 7 to 14 minutes of incubation. Then lastly, a very important test that we do is what we call the Benedict's test, and it's based on the principle of determining the amount of reducing sugar that you have in your sample. Remember, starch can be hydrolyzed to form maltose, so the reducing sugar for starch is the maltose. Then to do this, what you do is you need to fill up 1/3 of the beaker with water, and you put it in you heat it up to the boiling point, and you allow it to boil for a while. And while you're doing that, you get a clean of test tubes, like 5 of them. You label them, and then you add 5 ml of Benedict solution. And then after doing this, you add 8 drops of your sample to each of the test tubes that you've labeled, and you take your samples, you put them in the boiling water, and you observe for color change. So if you have an orange to the brick red, brown precipitate or coloration being formed, it means that you have reducing sugar in your sample. So the amount of coloration, the changes in the color that you have from green to red, orange to brick red, will tell you the degree of the reducing sugar, or the amount of reducing sugar that you have in your sample. So this is a demonstration of the IKI test, and you can already have your samples there. And once you had the drops of your IKI, you can see how the color is changing, and the color will reflect if you have starch in it or not. Like the 2A and a 3A, you can see that the color is blue black, which tells you that there's starch in your samples compared to the rest of your samples. And you can see the samples before I added the IKI. You can see with just your sample inside it, and then after adding the IKI, you can see the differences in the color even between the different samples. The 2A and 3A looks as if they still have the starch in them, and it means that there is no hydrolysis because the starch is supposed to be hydrolyzed if you have your amylase acting on them. Then the 4A still retains the color of the IKI, which means that there might not be any starch in the sample, which is why you have the iodine color there. Then the 1A and 5A looks as if they must have been hydrolyzed, and that's why you have the coloration, which is different from the rest of your samples. This table is already attached it to the module, Lab 7 module, and you can see the data sheets, and you can download it and attach it to your lab report. Make sure you fill up the tests and then the observation from the experiment and also the inference. The same thing for the Barfoed test. Before is you can see that the tubes look bluish in color, and then after before 15 minutes after, you could see the changes in the test tubes, especially 1 compared to 5, which is a positive control. The same thing, you need to fill up the forms and then uh, attach it to your lab. So lastly, for the carbohydrate digestion test, we use the Benedict's test with Benedict's solution. And this is the Benedict solution, and you did well from the protocol. And after boiling, this was before boiling, and after boiling, you see the precipitation that are formed around here. And the amount of precipitation or the color of the solution gives you the degree of the reducing sugar that is present in your sample. Remember, this is a positive control, and this is very similar to what you have here. You can see that this is actually contains a lot of the reducing sugar, and this contains a little bit of the precipitate here, the brick red precipitates here, and it should be similar to what you have here. Why these guys here are negative for reducing sugar. All right, you fill this form again, put the inference and everything. Then the second experiment is the protein digestion. BAPNA is the substrate, and I told you there is a synthetic protein. You have the enzymes, which is the trypsin. It should be present in the pancreatic extract or juice. And then we follow the same experiment as I described in the video for the protocols. And your vortex, remember the 5P is the positive control, the 1P to 4P would be your test samples. And then your vortex, you put cover slip, and then you put the 2P in the ice bath in a different temperature, you to see the effect of temperature on enzymes, and then the remaining one will be in 38-degree centigrade water bath for 60 minutes. And after that, you're going to record the color changes. So one thing you need to notice in this case is that if it's clear in color when you bring them out, when it's going to tell you that there's no hydrolysis, so it's negative for BAPNA hydrolysis. But if you see a yellowish coloration, it means that it's positive for BAPNA hydrolysis, and the intensity of the yellow color will tell you the level or the degree of hydrolysis. After this, we're supposed to do like a spectrophotometer to measure the degree of coloration that is formed, and we get like the optical density, and we can plot that. But because of the situation on campus, so there's a little bit of restriction, so I couldn't perform this side of the experiment. So you can ignore this part, but you have the difference in the coloration, then you're supposed to record that like 1P and 5P here before incubation, 1P, 2P, 3P, and 4P are like almost clear in color compared to 5P, which is yellowish in color. But when you look at the after 1 hour of incubation, you can see this looks yellow in color, similar to what you see in the 5P, but these other ones appear to be negative. All right, so the same thing, you need to fill this data sheet form and attach to your lab report. Then lastly, we have the lipid digestion experiment. The substrate is the milk or litmus cream. You have the enzyme to be lipase, and it's supposed to be present in the pancreatic juice. And then you have the emulsifying agent, which is the bile salts, and like I said, is very important for lipase digestion because it increases surface area and breaks the fat globules for lipase to be able to act. better. All right, so you have 6 tubes, and you're supposed to label them 1L to 6L and add all these solutions to them, like I mentioned in the video for the protocols. And 6L will be your positive control. And remember, you need to compare every of your samples with your positive control, and you tell me the differences in the color. And remember that the promotimer blue is going to be like a standard because fat digestion is based on the changes in the pH, and the promotimer blue is at 7.2 pH, so you need to regulate the pH for these samples before incubation to 7.2. And then we are going to compare that after incubation to see the changes in incubation and compare with the promotimer blue standard. And for the results, we are supposed to have, if it's blue, it's telling you there's no fat hydrolysis, and if it's yellow, it's telling you that there's a fat hydrolysis. So the intensity of the yellow color will tell you the degree of hydrolysis that you have in your sample. So I have a standard that I use to estimate the pH changes, and also we can use a pH indicator, which I also use. So what I'm trying to show you now is how bromothymol is used as a pH standard indicator for the lipid digestion test. So you see where you had 2 drops of bromothymol to each of your test tube, it turns it to blue color, and that color is supposed to be close to the bromothymol standard at a pH of 7.2. So what you do is after you vortex it and make sure that the color is close to what you have for 7.2, so you have to compare with the standard. So if it's more bluish than the standard, it means that it's at an higher pH, and what you do is to add HCl to it to reduce the pH. But if it's less than like it's kind of lighter in color, then you have to add NaOH, which is a sodium hydroxide, to increase this the pH to 7.2. So I did that, and after I'm done with that, then I have to do the incubation so that I can measure the changes in the pH. So this is what you're looking at here now is the difference between before incubation and after incubation. You can see here, this is the standard, and the pH looks almost 7.2 compared to what I have for the bromothymol standard. And then there here above here is the different pH standard for bromothymol, and you can see the less the pH, the more the yellowish it becomes, and then the higher the pH or alkaline, then the more bluish it becomes. And the same thing here, when you look at after the incubation, you could see that IL is turning yellowish in color, which shows you that there's some form of hydrolysis taking place there. 2L, 3L, and 4L still retains their bluish coloration, and in fact it's kind of closer to what you have here compared to the 5L and the 6L, which has as our control, as our positive control. So in your result, I'm expecting you to tell me the reasons why 1L, 5L has a slightly similar correlation to 6L, but 2L, 3L, and 4L doesn't have it. So you record your observation, your test here, what you added together, then your observation, the changes in color, and then the inference is going to tell me what's going on, is there hydrolysis or no hydrolysis, and what pH can you estimate for each of the tubes.